So far we have talked about how the action potential coming to the presynaptic terminal is going to lead to the opening of the calcium voltage-gated channels, and that calcium is going to enter. What we are going to now learn, how is this entering of the calcium going to lead to the fusion of the synaptic vesicles with the plasma membrane and the release of neurotransmitters. So, as you can see here from these two models of synaptic vesicles in the active zones, molecular machinery of exocytosis is really complicated. There are many proteins in synaptic vesicles, and there are many proteins at the active zone that play a role in this process. We're going to be mentioning just some of these proteins. So what is active zone? Active zone is this electron dense and it's very biochemically insoluble material which is located presynaptic plasma membrane, and it enables the tight coupling of neurotransmitter release to an action potential, so it's right across from the receptor or postsynaptic membrane, and it directs the release of neurotransmitters to the synaptic cleft. What active zone does is several important steps. It recruits vesicles. It has several different pools that we will be discussing more in the class. Then it docks the vesicles, so these are available vesicles. Then we have vesicles that are docked at the active zone, then they're primed, and then when calcium enters, they can fuse with the membrane, and then they're retrieved through the process of endocytosis. Process of endocytosis we will be discussing in next lecture. What we are going to discuss today is docking, priming, and fusion. So here is some of the molecules of the active zone. Again, we are not talking about all of them. We are not trying to cover them extensively. What you should know are several proteins such as RIM, which stands from Rab interacting molecules. So RABs are small molecules, GTP proteins on the vesicles. So RIM interact with them, but they also interact with the calcium channels. So they will guide or prime the vesicles, but they're also involved in tethering calcium channels next to the docking site. So this is a common theme, that lots of these proteins in the active zone are going to do several different things, and therefore the null mutants that are usually used to study their role will have several different phenotypes, and one may mask the other one. Apart from RIM, we have RIM binding proteins. Somewhere they will be referred as RBPs, which are also involved in docking, in tethering calcium channels to the docking site next to the vesicles. In addition to these, we have the proteins such as Piccola and Bassoon, which will guide the synaptic vesicles to the active zone. In addition, Bassoon, for example, is also known to tether these calcium channels through the interaction with the RIM binding proteins. We also have a very important proteins called MUNC13, which transforms synaptic vesicles into the vesicle in a fusion component primes. So they are very important in vesicle priming, which we'll soon see what it is. And we also have the protein that such as liprins and ALKs, which are really important to precise localization between pre and postsynaptic membranes. And we know that netrins and neuraxins are proteins that also play a very important role in the pre and postsynaptic membrane interaction. So what I would like you to remember is just that, again, we have the molecules that are important tethering calcium channels right next to the synaptic vesicles, because as if you remember from the previous lecture, we really want the calcium be really close to the synaptic vesicles that need to be fusing. So there is, again, on the synaptic vesicle, we also have the protein network. Similarly, as you have seen, very complicated version. This is much simplified version. Here you have several different proteins. We have synapsins, which we now know they're really abundant. That's how they got the name. Lots of these proteins were originally identified with biochemical purifications of presynaptic compartment and then vesicle compartments, and then afterwards they used genetic cloning in order to find what their roles are. So for synapsin, we know that they play a role in trafficking of the vesicle, especially in recruitment from the readily available pool to the ones that are going to be docked and primed and released immediately. Then we have Rab proteins. As I mentioned, these are GTP binding proteins that are involved in docking. As I said, interact with MUNC13 and RIM at the active zone. So therefore they play a role in tethering synaptic vesicles or docking them right close to the calcium channels. We have synaptotagmin, which is a calcium sensor for exocytosis. We'll talk a little bit more about it. It has 2 calcium binding sites and it can bind a total of 5 calcium ions. Then we have very important snare proteins. Synaptobrevin is on the synaptic vesicle, while we have 2 other snare proteins such as SNAP25 and cetaxin, which are on the plasma membrane. They together form snare complex, which is crucial for the fusion, which is very important in fusion. And then we also have neurotransmitter transporter, which we will be talking in the part when we cover endocytosis and then transfer neurotransmitters into the vesicle again. As I said, fusion is mediated by snare complex. Snare is short for soluble and ethyl-melamine sensitive factor attachment receptors. These proteins are universally involved in fusion, so when you have any membrane fusion inside, so trafficking of the protein through Golgi to endoplasmic reticulum, it's going to involve the snare complex fusion. They are characterized by this snare motif, which is around 60 residue long conserved sequence and makes these alpha helixes. So every time when you have vesicle, which is going to fuse with the membrane, so you have vesicle snares, which are otherwise called as V-snare, and in this case it's going to be synaptobrevin. So different synaptic vesicles will each have its own synaptobrevin. They're different synaptobrevin molecules or V-snares. And then target membrane snares are going to be called T-snares. In this case there are 2 of them, syntaxin and SNAP25. So synaptobrevin and cetaxin each have 1 snare motif, but SNAP25 has 2 snare motifs, which then assemble into this 3-helix bundle, which is really tightly coiled. And this snare complex, that coiling of it, is what actually draws the 2 membranes, membrane of the vesicle and the plasma membrane, close together. So this is a vesicle docking. Vesicle comes, we again have synaptobrevin on the plasma and synaptic vesicles, and we have SNAP25 and cetaxin in the plasma membrane. When they form this snare complex, this pulls those 2 membranes very close together. And actually, this formation of the snare complex can catalyze fusion in vitro, but it's much slower and less efficient, and there is no regulation by calcium, which we know in the neurons that the fusion of the vesicle with the plasma membrane is going to occur only when the calcium enters. And again, here we again have synaptotagmin, which is our calcium sensor at the synaptic vesicles. So again, this is a little bit better shown, this vesicle priming of this snare complex, how closely it pulls together these 2 membranes. So the lipids form stable complexes, they don't like to fuse. So it's though the energy released by forming the snare complex between synaptobrevin, cetaxin, SNAP25 is going to provide the energy to pull them very close together. And again, here is synaptotagmin with 2 calcium binding sites. On 1 calcium it can bind 3 calcium ions, on the other binding site can bind 2 calcium ions. So here is the whole sequence of events. Again, this is what we have seen. We have vesicle that docks, that snare complex forms and then pulls these membranes close together. Then calcium enters, binds synaptotagmin. When calcium binds synaptotagmin, it increases its binding to phospholipids. So now the synaptotagmin can also insert itself into the plasma membrane, and this is enough to catalyze the membrane fusion of the vesicle and the plasma membrane. One useful tool that everybody's using to study the fusion and chemical transmission is using tetanus toxin and botulinum toxins. How they work, why do they prevent synaptic vesicle release, is because these are the toxins, these are enzymes produced by bacteria, which actually will cut these proteins. So they're cutting the snare proteins. When they cut the snare proteins, you cannot form the snare complex and there will be no vesicle fusions. So often you will see mice in which you can express tetanus toxin in specific cell type to show the effect of preventing synaptic vesicle release from those cells. And again, here is this, again, botulinum toxins and tetanus toxins cut snare complexes in several different places. After the fusion occurs, this complex has to be now dissociated. There is a complex called NSF and SNAP, which come and then they dissociate the snare complex in ATP dependent manner. So they need energy of ATP. So what ends up happening, then you have dissociation of snare complex and you have the membrane snare, SNAP, and 25 acetoxin at the plasma membrane, while we have synaptobrevin or vesicle snares dissociated. So this is what we know about biochemically and through the studies of what happens with the mutations. How this exactly looks at the synapses or active zones is much less known. There are some really cool microscopy studies. This is an example of electron microscopic tomography by Harlow, 2001, and again from neuromuscular junction, in which you have synaptic vesicles which are tethered to the channel. So we have these ribs and beams, that's how they call, that's how they really look. The identity is really not known. These are probably RIM and MUNC13 proteins. The PEGs that I see are calcium channels, but again, identity of these structures is much less known. It needs to be confirmed.